Okay, so in the last video we talked about these innate defenses, and now we are switching gears and moving over to the adaptive or specific defenses, right? And this is where true immunity comes into play. So this section I would say is complicated, but hopefully we can kind of lay it out in a way that makes sense. So what we're going to see as we dive in here, just like a brief overview here, is that our lymphocytes, our B cells and our T cells, are always acting in response to an antigen. So an antigen, typically it's a protein, right? But it's a surface molecule. We have lots of surface molecules, but you wouldn't normally call them an antigen because the antigen should be that surface molecule that actually stimulates this immune response, a response by B lymphocytes or T lymphocytes. Now, one of the things that's super important in how this works is that that all lymphocytes use antigen receptors. So they have a protein on the surface of their cell that's very specifically shaped, and kind of this lock and key mechanism. There's only one antigen that it should fit, right? And that's how we actually have this specificity. That's how we react to these pathogens. Okay, so let's start digging in here a little bit. Okay, so again, just a reminder now that we're in lymphocytes that both T cells and B cells start in the bone marrow, right? But some of these cells are going to migrate to the thymus where they mature into T cells, right? Others remain in the bone marrow and become B cells, or remember the NK cells, how I said they were cousins, right? So they're just over, they mature in the bone marrow as well. Okay, but what we end up seeing is T cells, B cells, and natural killer cells all moving out into the tissues to create their own unique response. The big difference between, and same thing from your book, the big difference, right, between B cells and T cells is how they respond. So T cells are responsible for what we call cell-mediated immunity. So T cells, particularly a group of T cells called cytotoxic T cells, are going to be looking for pathogens, but pathogens often that are already in our own cells, something like a virus, right, or a mutation that might cause something like cancer. The antibody mediated immunity, which is sometimes called humoral immunity. And remember, humoral is just like an old way to say the body fluids. So humoral immunity is through antibodies. So B lymphocytes give rise to these antibodies that are then going to circulate through the blood. And either way, right, we're working on destroying that pathogen. Come on. Okay, so this is the antigen receptor. Again, every B cell, every T cell is using an antigen receptor. And what they're trying to show you here is that these proteins are essentially all the same, right? Every antigen receptor across B cells, T cells are the same, except for this, what's called the variable region, and that includes the antigen binding site. And so you're going to have slight differences in the shape of this binding site that leads to specificity. Now, the production of this protein, like every other, right, antigen receptor is a protein, the production of this protein is regulated by DNA. So what you end up doing during development is you have actually multiple segments of DNA that help code for these receptor sites. But what you end up doing is like randomly grabbing a piece from here, here, and here, and that makes one shape. And then you grab from here, here, and here, and make a slightly different shape. And so it's this kind of maybe like recombination that you could think of that gives us a huge diversity of antigen receptor shapes, right? Millions, maybe even billions of different antigen receptor shapes. Each lymphocyte is only going to have one receptor shape, right? It might have hundreds of antigen receptors on its surface, but they're all identical, right? And then the next lymphocyte has a slightly different shape, and on and on. And so one of the things that is key in this immune system working is that we only want antigen receptors to foreign proteins. You do not want antigen receptors for any of your self proteins, right? You don't want a sodium potassium pump causing white blood cells to start attacking cells, for example. And so what's going to happen, this is one of the really important things that actually happens in the thymus. During development, you run this screening process. And so you start showing the lymphocytes all these different proteins from the body. And if any of them are like, woo, an attack, you go ahead, the thymus goes ahead and destroys that lymphocyte, right? So the other thing to keep in mind is you may only have one lymphocyte with the correct antigen receptor shape. And it's kind of silly to think that one lymphocyte could fight off like a bacterial infection, right? Because that bacteria is multiplying rapidly. And so what we're going to see is that what we need to do is basically figure out which lymphocyte actually matches this particular threat and then start creating clones of it. Build a whole army of clones so that they have that same receptor site as well. This is, okay, it's pretty cool. All right, so another thing that ends up being really important in understanding how this immune system works is what are called MHC proteins, or major histocompatibility complex proteins, MHC. Another name for those is HLA, human leukocyte antigen. I'm sorry that it has multiple names. Typically, I've been seeing a lot of people refer to it as MHC. So these MHC, major histocompatibility complex, tissue, remember histology is the study of tissues. So this deals with tissue compatibility. These proteins were really discovered as we started doing organ and tissue transplants, because you need more than just blood type. In order to have a successful transplant, you need to have these proteins look as much like each other as possible, right? To have a good match on a donor. So what we're looking at here is there are two different types of MHC proteins. Let me get my... Nope, can't oh, here we go. We can annotate, right? So notice up here, they're talking about MHC class 1, right? Or MHC1. These are going to, this class of proteins, separate from class 2, down in this picture. MHC1s are found in all nucleated cells in the body. So basically everything except red blood cells, right? And the way this works is an MHC is a protein that your DNA codes for it, and you're going to make it pretty consistently. But what we do is, and that's what this big blue molecule here is, right, is this MHC. But what we do with the MHC is we take samples of all the other proteins that are being produced in the cell. So remember, proteins are produced by ribosomes. So, sorry, MHC1 is taking a sample of every other protein that's made by a cell. Remember, proteins are made at ribosomes, and any protein that is bound for export actually is going to go through the endoplasmic reticulum and then the Golgi apparatus, where it's packaged into a vesicle and goes to the surface. So MHC1 proteins are just picking up samples of other proteins and carrying them to the surface. They present them to parts of the immune system, right? In particular, what we're going to see is these MHC1 proteins are going to be checked by cytotoxic T cells. Okay, so every cell in the body is constantly taking a sample with MHC1 and putting it up on the surface for the immune system to check. And this is brilliant. So this works really nicely if you, say, had a mutation in your DNA. You would make a different protein, and that protein would get carried to the surface. A mutation in your DNA might lead to cancer. And so now you carry this abnormal protein to the surface of the cell, and your immune system actually recognizes it as foreign. A cytotoxic T cell should kill that cell, right? Nipping cancer in the bud. The other place this really comes in handy is what they're showing in this cell, that it's been infected with a virus. Right? And so that viral nucleic acid is now producing proteins, right? The cell is producing these proteins for a virus, but the MHC1 still is taking samples and it carries it up to the surface, right? And cytotoxic T cells should notice that this cell has been infected and again, kill it, cutting off the ability of that virus to replicate. All right, down here, when we look at MHC2 proteins, right, these are only going to be on what are called antigen presenting cells. So this is going to be, so notice it says a phagocytic antigen presenting cell. Basically, this is going to be things like macrophages, dendritic cells. We'll also see B cells can use this. So what's happening here is class 2 MHC proteins aren't really interested in gathering our own proteins and carrying them to the surface. But remember, phagocytic cells, when they engulf, say, this bacteria, they actually hook it to a lysozyme, which is where all those enzymes and nasty things are, and grind it up. The MHC2 protein is going to take a sample from that vacuole, from that engulfed, destroyed microbe, and it's going to carry it to the surface, again, to present it to the immune system. And what we'll see here is typically it's going to be helper T cells that are checking these antigen presenting cells. So a helper T cell docks on this class 2 MHC protein. Okay, so figures from your book, again, trying to show MHC class 1, taking a sample of every protein that's made inside of your cell, right, and holding it on the surface. That can be a foreign protein, right, if we've been infected by a virus, or it could be a self protein, right, because everything is getting carried to the surface. The foreign antigen should cause an immune response. The self protein should not. Okay. This is then your textbooks showing of how an antigen presenting cell works. So again, our macrophage, we've gone ahead and engulfed a bacteria, ground it up, and here we see the class 2 MHC proteins are going to take a sample to the surface, and of course that's foreign, and so the immune system should be stimulated by that. This is, I don't know, to me, this is like the immunity, this adaptive immune response is just like the coolest thing that our body is able to pull this off. Okay, so here's what this would look like, right? So here we have, in this case, an infected cell, right? We've gotten a virus or a bacteria inside here. And so again, if this is just like a respiratory cell, right? Maybe this is SARS-CoV-2 infecting our cell here. I'll get out of your way. Right? A sample of it should be carried to the surface by a class 1 MHC protein, right? Because it's on a self cell, just a normal nucleated cell. What should be checking then is a CD8 cell. This is another way to say a cytotoxic T cell. Let's zoom in here just a little bit, right? So here's our class 1 MHC, right? So big blue protein, it's holding our antigen. And what we need to have happen here in order for a cytotoxic T cell to work, right? It has what's called a CD8 protein. The CD8 protein actually binds here to the MHC1. That's how they recognize what they're checking. But then that antigen receptor on the surface of that cytotoxic T cell needs to match the antigen, right, that's being held on the surface. Okay, so you could actually have multiple cytotoxic T cells come by and not have the correct receptor shape. And so it's when you finally find that one that's the correct antigen receptor to say, wait, this is foreign, we should attack it. Right? And so what we're going to get here is that cytotoxic T cell is actually going to become activated, right? It gets triggered when we actually get the correct receptor matching that antigen. So what we have to do then is actually, this is where I was talking about making all of those clones, right? It took you a while to find the correct lymphocyte that matched, and now we want thousands of that same lymphocyte. And so this is going to be a process of cloning. So they're saying cell division, right, but making identical copies. Notice that some of those T cells are active And a cytotoxic T cell, so T sub C is cytotoxic T cell, is going to basically blow up the infected cells, right? Starts shooting little holes in it and makes it lyse, which is kind of, man, poor cell just got infected with SARS-CoV-2 and then we're going to explode it. But it's taking one for the team. If we can get rid of that cell that's been infected by a virus and prevent 10,000 copies of a virus from coming out and infecting other cells, then it's worth it, right? And so that's what this cytotoxic T cell is going to do. So here's the zoom in, right? We're using perforins, lymphotoxins, anything we can do to go ahead, cytokines, right, to go ahead and then destroy the infected cell. But notice some of those clones become memory cytotoxic T cells. They never become active. They're not, shooting all these chemicals into the infected cells. We are going to save those for later, right? This is like the National Guard. They didn't go over to fight the war. They're staying at home in case we see this same pathogen again. Then these memory cells are going to be ready. That's going to be their moment. Okay. So this is that idea of clonal selection. This is in B cells, but it works the same in T cells. Notice what they're showing you here is every lymphocyte has a slightly different receptor shape on its surface, but all the receptors on the same lymphocyte are the same. And so when an antigen is introduced, we need to go through and find the chosen one, right? That one lymphocyte that has the correct receptor shape to match the antigen. We're going to then ask it to undergo, so that's clonal selection, is picking the correct one. And then we undergo clonal division, where we start making those clones, or clonal expansion, same as clonal division. And again, some of the cells actually become active and others remain as memory cells. Get it? Okay, so that's cytotoxic T cells that we were looking at. Here we're moving over to helper T cells. Now, helper T cells, remember, are going to be looking at antigen-presenting cells, so our macrophages and our dendrites. So here, our antigen-presenting cell, right, we're in the antigen-presenting cell, uses a class 2 to hold that foreign antigen on the surface. The helper T cell, right, same thing, we need to make sure we have the correct antigen receptor. And so it can take a while, right, to have the correct antigen receptor bind here. But notice what allows helper T cells to check MHC2s is what's called the CD4 protein, right? So the CD4 protein is really what kind of docks here on that MHC2. And so sometimes you'll see helper T cells called CD4 cells. Now, the way I keep CD4 and CD8 straight is I think that helper T cells, they call for help, right? So they have a CD4 protein, if it works for you, where cytotoxic T cells use the CD8. Okay. Same thing is going to happen here, right? When we find the correct helper T cell, right, that actually bound there, we're going to go ahead and do clonal expansion. So we start making lots of clones. We keep some as memory cells for next time as well. Now notice what the helper T cells are doing. So they're showing cytokine, cytokine, cytokine. This is a chemical messenger. And basically what it does, what helper T cell cytokines do, is they stimulate cytotoxic T cells to be more lethal, right, to be more active. And they also stimulate B cells to be more active, start producing more antibodies. So helper T cells really talk to both cytotoxic and B cells. They're really this like linchpin in the immune system. If you don't have helper T cells, you can basically, your immune system never really gets ramped up and goes after these pathogens. That's what makes HIV so bad, because HIV actually infects these helper T cells, and then they're not multiplying, they're not sending out these cytokines, and so the rest of the immune system essentially is non-functional. Okay, so we still have to look at, okay, same thing, antigen-presenting cell being checked by a helper T cell. B cells is who we have left here. So it works very similarly. So here we see the B cell, right? It has antigen receptors on the surface. It's found this foreign antigen, and it's going to go ahead and tell a helper T cell as well. So the B cell doesn't do phagocytosis, but it can process the antigen, hold it on the surface on an MHC2 protein. Okay, MHC2s dock with CD4s on helper T cells, and so our helper T cell can go ahead and clone itself, be activated, send out cytokines, and here we go, interleukins, another cell messenger. Notice this is going to back to the B cell, right, that just showed the antigen to the helper T cell. These interleukins are going to tell this B cell, you're the chosen one. You have the correct receptor shape. Only you can do this job, right? And so here we start to see that cloning process, right? And again, they're showing you two, but this is by like the millions, right? Cloning, cloning, cloning. Again, some of our B cells are going to stay as memory cells for the next infection, but some of our B cells are going to turn into what are called plasma cells. So the plasma cell is basically an activated B cell that's capable of producing antibodies. Antibodies are proteins that basically are the same shape as the antigen receptor, right? Because if this antigen receptor right here had the perfect shape to match the antigen, now we're going to make a bunch of copies essentially of that antigen receptor, but we're going to set them loose in the blood, and now they're called antibodies. So antibodies fall into a few different classes. You may see antibodies called immunoglobulins, which is why then you'll also see this abbreviation, right? IgG, immunoglobulin G. So we come in basically 5 different categories of antibodies. They all have slightly different roles. Probably not super important to memorize all of these details at this point, but it can be useful to know things like, so IgE, for example, IgE binds to mast cells and causes them to release histamine. So typically, if you're dealing with someone with allergies, they are producing a bunch of IgE, maybe overproducing, right? We often refer to allergies as a hypersensitivity of the immune system, producing way too much IgE, activating all these mast cells, releasing all this histamine. Next thing you know, your eyes knows they're running because you caused all this vasodilation, right? IgE, it might be important to note that, IgA is the most common antibody found on mucous membranes. So it's in things like saliva and tears, on all these mucous membranes. It actually crosses to a baby through breast milk. And so it's going to be really important in preventing infections of those mucous membranes. So people who maybe don't make as much IgA are more likely to develop respiratory tract infections, pneumonia, things like that. IgG, you might notice, is by far, right, the most common antibody that we produce. This one actually crosses the placenta. So baby's actually born with some protection, just being handed off from mom. And then, of course, in breast milk, you can get the IgA. In IgM, one of the things that I think you might want to know about that one is that it's often this first antibody produced. So you have lots of circulating IgG, but then when you see antibodies production increasing in response to an infection, it's IgM that's kind of that first one that jumps in there. Okay, so different categories, but notice all of them, they have that same antigen receptor shape at the end, though IgM is kind of crazy, right? It's actually a pentamer. It's got 5 different ones hanging off the outside. Sorry, the dog is barking. Okay, so one thing that I noticed that students have a hard time with, this is good, right? So when we start producing antibodies, notice this is like where you can see immunity. So the first infection, right? Notice there's this like lag time where we're basically doing clonal selection, clonal expansion, and then antibody production, right? And so we get this peak in antibodies, and it's once you produce enough antibodies that you really can start clearing the infection. And so this period of time, you're sick. The secondary infection, the second time you come in contact with the same antigen, the same pathogen, all of those memory cells that we've been mentioning just boom, immediately are able to respond, right? And so we don't see that lag time, and we see a much more dramatic peak in antibody production. And so this is immunity, right? There's no time to be sick in this one, right? Your body takes care of it before it can actually cause disease. Your book here has figure 2221, I thought did a really nice job of showing how that works, right? So in the first exposure, this can take weeks, right, to go through this whole process of clonal selection, division, right, making memory cells, making plasma cells, making antibodies, right? But you can clear the infection. But next time, notice it's a much shorter response, hours or days to take those memory cells, right? Make tons of clones, tons of plasma cells, tons of antibodies. And so we actually see a much stronger secondary response. And you don't get sick. That is the basis of immunity. Having those memory cells, having immunological memory means that you can respond to that same pathogen much quicker and before you get ill. Okay, so this slide I thought was a nice summary. So, before I go to this, step back here to antibodies with me. So one of the things that I find that students often kind of miss is it's like, well, what is this antibody actually doing, right? So you said it looks like an antigen receptor, now it's floating through the blood. So it is going to seek and stick to those antigens. So if the antigens are all over, say, the surface of a bacteria, what you're going to end up with are a few different effects. So if antibodies, say, cover all those antigens, it may neutralize that microbe, right? Maybe that protein was actually important for how the cell produces toxins or docks to a host cell or something like that, right? So by coating it in antibodies, you can actually neutralize it. You can use antibodies in a way that they stick a bunch of the pathogen together. And so with that pathogen maybe was circulating or moving freely through the body, and now you're doing agglutination, you're like sticking it together. Often that'll, if it was in the bloodstream, that would cause precipitation. It would fall out of circulation. You can use antibodies as tags to actually attract phagocytic cells. So phagocytes are like, that has an antibody on it. It must be foreign, and it's going to go attack it. They also do what's called opsonization. So picture that same bacteria covered in antibodies. It's actually much easier for the macrophage to engulf it, right? The bacteria is not slippery and smooth anymore. It's got all these antibodies on the outside. So opsonization is this increased effectiveness of phagocytosis. So those sorts of things, right? So antibodies physically are attaching to those foreign antigens on our pathogen and doing something: neutralization, agglutination, attracting phagocytes, opsonization, etc. Okay, now we can go to the review slide. So this is kind of a nice overall review. So let me I got the pen. Okay, so again, notice up here they're talking about innate immunity, right? Non-specific, so we got our physical barriers, our phagocytic cells, some of those chemical mediators like interferons, the inflammatory response, fever, etc. Okay. But then we move into adaptive immunity. So one of our macrophages in this case, come back. One of our macrophages in this case engulfed the bacteria, becomes an antigen-presenting cell, and is showing it to a helper T cell. That helper T cell clones itself, proliferates, starts dumping a bunch of cytokines that activate B cells and activate cytotoxic T cells, right? So now our B cells are going through clonal selection. Our B cells are producing antibodies, right? Our cytotoxic T cell cloning itself, becoming more active, it's going to attack, right? So this is how our body defends itself. So notice there are connections here between innate and adaptive, right? We see that macrophage acting. We might see things like, remember our antibodies, we said could actually complement complements the effect of antibodies. So if antibodies attach to something, we can increase or improve phagocytosis. We can cause complement to create that membrane attack complex. So these are related. Okay. So then again, that's the kind of the summary from my previous textbook, same thing. From a pathophysiology textbook, this one I like because it reinforces this idea. So here's our antigen. The macrophage becomes an antigen presenting cell. The helper T cell stimulates both sides, the B cells and the T cells, right? And then we're going to go ahead and attack the B cells, become plasma cells, create antibodies. Our cytotoxic T cells are activated, right? And we're going to attack. What it's showing here is on the secondary exposure, notice how much quicker this all happens. All the arrows are shorter and boom, right? Our T cells and B cells take care of it. That's immunity. The memory cells being present, quickly becoming activated. Okay, last thing I wanted to mention, right? When we talk about adaptive immunity, is there's a couple different ways that you can have immunity. So it can be either active, meaning your own immune system has created memory cells and you have immunity, or it can be passive. And this basically means it's being passed on to you by another person or animal, typically in the form of already made, right, preformed antibodies. So you haven't mounted an immune response, you don't have memory cells, but we can hand you antibodies. So let's look at what these look like. So active immunity, your own immune system having to create this response, it's either natural, right? You come in contact with a virus, a bacteria, you mount a response, you're sick, but once you get over it, you have immunity, right? But we've come up with ways, humans, so clever, of artificially stimulating that immunity. This would be vaccination, right? So you deliberately introduce a little antigen, right? A little piece of that virus without the whole virus that might make you sick, right? There's a few different ways to do it, but you stimulate the immune response, you make memory cells, and so then if you ever do come in contact with that pathogen, you're already ready for it. Passive immunity, naturally, these are going to be the antibodies that are passing from mom to baby through the placenta or breast milk, right? And so that baby hasn't come in contact with all these diseases, but it has some defenses present, right? So it doesn't need to get sick when it's so little and vulnerable. Artificially, this is kind of crazy. This has been in the news a lot, right? Some talk about using convalescent plasma. So someone who's gotten better from COVID-19 has the antibodies circulating in their blood plasma. We can actually collect that and give it to someone who's sick to see if it helps them. And preliminary studies says it helps. It doesn't seem quite as strong. We've used this before in things like Ebola as a pretty effective treatment. I haven't seen great studies here on COVID-19, right? But it's promising. You can also do this. You could use an animal. So for example, you can infect a rabbit or something with rabies, cause the rabbit to produce antibodies, and then you can steal the antibodies from the plasma of that rabbit. So sometimes we can use preformed antibodies that were actually produced by a different animal. So again, to give us some protection until our own immune system can ramp up and protect us. All right. That is it. We'll see you guys in class.